So I'm heading the National Reference Center at the Institut Pasteur in Paris, and our main mission is the surveillance of the disease in mainland France, but also in French overseas territories. So since probably most of you are not very familiar with the disease and the pathogens, so Leptospirosis, Leptospira belong to the family of is a bacteria, and belong to the phylum of spirochetes. And within this phylum of spirochetes, so you will find several major pathogens, such as Treponema pallidum, the agent of syphilis, Borrelia burgdorferi, the agent of Lyme disease, and Leptospira interrogans, the agent of leptospirosis. As you can see here on this, in the different pictures, all these spirochetes, they share the same morphological features, so they are very thin and helical shaped bacteria. And as you can see here, but they also have endoflagella, so flagella that are not external to the bacteria, to the cell, but within the periplasmic space, and that makes these bacteria highly motile in highly viscous media. So, brief history of leptospirosis. So the first description of the description of the agent of leptospirosis was by a Japanese group almost 100 years ago. And what they did in this first publication is they inoculate the blood of a patient with what was called at that time the Weil disease. So they inoculated hamsters with the blood of these patients, and these hamsters, infected hamsters, died of leptospirosis, and they find in the organ of these infected hamsters these bacteria that you can see here with this typical morphology. So the same group in Japan published several papers at the same period, so more than 100 years ago, and they have shown that so these bacteria are the agent of leptospirosis. They also have shown that the rats are the main reservoir, and they also have shown that water is probably, exposure to water is probably a major route of transmission of the disease. And so these are really landmark papers, studies, and we did not learn so much on the disease since these first publications. So the cycle of leptospirosis. So leptospirosis is a zoonotic disease. So two groups of animals can carry the pathogenic bacteria. The rats, which are asymptomatic carriers of the bacteria, and every other mammal, so wild animals or domestic animals. And in this case, these infected mammals can develop the disease. So all these animals can carry the bacteria in the kidney, in the renal tubules, and they are going to excrete the bacteria in their urine and then contaminate the environment. So the main route of transmission to human is the indirect route of transmission. It's the contact of the water contaminated by the urine of these animals with the abraded skin or the mucous membranes. Then you have dissemination of the bacteria in the host, in the bloodstream, and the bacteria is going to rapidly reach target organs such as the kidney or liver and all organs. And so in the subset of patients are going to develop severe disease with hepatic and renal dysfunctions that can lead to death. So the main problem for leptospirosis is the diagnostic. So as you can see, there's no specific, there's a list of other diseases that mimic leptospirosis, so there's no specific symptoms. It's an acute fever, it's a flu-like syndrome, and which makes the clinical diagnosis difficult, and also the biological diagnostic is difficult. We don't have very efficient tools for the diagnostic of the disease. So about the burden of the disease, so there was a recent study who estimated to more than 1 million severe cases of leptospirosis worldwide and 60,000 deaths. As you can see here in this map, most of the most prevalent, the countries with the highest incidence are in tropical countries. So in Southeast Asia, for example, in the West Indies, in Latin America, etc., etc. And I added here a question mark for the African continent because we have very few studies on the burden of the disease in Africa, in most of the African countries. So just briefly about Africa. So here on this map you can see the countries where leptospirosis have been reported in humans or in animals. So the countries in white are where the disease has never been reported. It's not because the disease is not there, it's just because nobody really look at the presence of the pathogen in these countries. And here another study in Tanzania showing that a significant proportion of non-malaria cases can be cases of leptospirosis. So this is true in Tanzania, but it's probably true in many other countries in Africa and also in Asia. So leptospirosis is probably under-recognized, is probably an under-recognized cause of acute fevers in many tropical countries. So leptospirosis is a neglected disease because, as I just told you, we have limited information on the disease burden, because it affects the most neglected and marginalized populations, because we lack adequate diagnostics, there's no effective control measures. As I told you, the main reservoir are rats, and it's almost impossible to get rid of the rats, of the population of rats. There's no universal vaccines available, so Emaxio is commercializing a human vaccine, but this vaccine is not available worldwide. And there are a few vaccines that are also available for animals, but these vaccines do not allow cross-protection against all the serovars that are circulating. And we have finally a limited understanding of the epidemiology and the pathogenesis of the disease. So here, just to show you the list of the priority neglected tropical diseases as established by WHO. So this list is regularly updated by WHO. And so today, I think this list was updated last year in 2017. And there are 17 pathogens listed here. And as you can see, leptospirosis is not among the list of WHO. So leptospirosis is a neglected disease, but it's highly neglected even by WHO. It's not considered as a neglected disease by WHO. So here it's a recent work that we have done with some other colleagues, just showing that leptospirosis is really neglected. So here you can see the research funding on different diseases, including neglected diseases from WHO here indicated by the green in green. And you can see that leptospirosis, so when you compare the research funding and the burden of the disease here in terms of DALI, you can see that leptospirosis is highly neglected, even compared to the other neglected diseases indicated in green. The same when you look at the H index, at the bibliometry, so you see that leptospirosis is also highly neglected among all these different diseases when you compare leptospirosis and other diseases with comparing the global burden, the relationship between the global burden and the H index. So leptospirosis for sure is neglected. So it's also an emerging disease. So this is due to the demographic and ecological changes. So today there are about 1 billion people living in slum areas, like in Brazil, for example, in the favelas, or in India. And in these slum areas, so every year during the rainy season, you have outbreaks of leptospirosis due to rat-borne transmission in these slum areas. And this human population living in slum areas are going to double in the next 25 years. So because of this demographic change, leptospirosis is probably going to emerge in the next few years. It's also emerging because of the climatic changes with the global warming and the more frequent appearance of extreme climatic events such as hurricanes that are responsible for floodings and outbreaks of leptospirosis in these tropical countries. Here, just to show you the strong association between heavy rainfall, the rainy season, and outbreaks of leptospirosis. So here it's an example in Mayotte, which is one of the French department, which is a very small island in the Indian Ocean between Madagascar and the African continent. And so you can see here in blue the monthly rainfall, and in the lane in orange the number of cases of leptospirosis every year. And you can see there's each year, after the rainy season, after the peak of pluviometry of the monthly rainfall, you have outbreaks of leptospirosis in this island. And this is you have the same picture in other tropical countries. So these are in developing countries, and in developed countries like here in Europe or in the US, for example, here. Water exposure also is strongly correlated, usually with cases of leptospirosis. So here it's an example of a triathlon in the US. So you can see on this scheme the day of the triathlon and all the confirmed cases and suspected cases for leptospirosis following this triathlon. And so you can determine, so here the box in black are the confirmed cases. So you can see the first confirmed cases happens about 5-6 days after the race of the triathlon. And so in this case, there was more than 50 participants of the triathlon with leptospirosis, which were leptospirosis cases. And this happens even here in Europe and in France every year. So here it's an example here in Brittany, in France, from 2 years ago. Among kayakers, so there was many cases, about more than 10 cases of people who got leptospirosis after doing kayaking in this river in Brittany. And so usually what happens is, so you have rodents or any other mammals that are going to contaminate the environment, the banks of river, for example, with their urine, and after heavy rainfalls or just rain events, the soil is going to be resuspended. All the particles of the soil, including the urine of these rodents, are going to be resuspended and contaminate the rivers. And then people then after are going to swim in this river, and then you can have transmission of the disease to these people. So what about the situation in France? So at the Institut Pasteur, we have a long history of passive surveillance of the disease. So here it's just a book from the first scientists who work on leptospirosis in France, so Louis Martin and Auguste Petit from 1919, so just after the First World War. So as you can see, so here the number of cases in mainland France in the last century. And what you can see here is that in the last 4 years, starting from 2014, we have a twofold increase of the number of cases in comparison to the previous years. So there's an increase, at least twofold increase of the number of cases. And this situation in mainland France is also observed in some of the European countries. So this is an example of Netherlands, also where there's a reference center for leptospirosis, and where there's passive surveillance for many, many years. And in 2014, they also observed a threefold increase of the number of human cases in comparison to the previous years. So we don't know exactly what are the reason for this emergence in Europe. So they can be due to the climate, winters that are mild winters that allow for the bacteria to survive for a longer period in the environment, to the increase of the rat population. It can be to people that are doing more water activities. It can be also due to the diagnostics. So there are more labs and hospitals doing diagnostics. So we don't really know what are the reasons of this emergence in most of the European countries. Just to show you some of the data of the reference center. So as you can see in the first scheme, so that the highest number of cases occur always at the end of summer, in August and September. So when there are probably more exposure to water during water activities. And below, so you can see the infecting serogroup. So I did not tell you, but there's a high diversity of serovar and serogroup among Leptospira. And so by using a serological technique called MIT, macroscopic agglutination test, which was developed 100 years ago at the Institut Pasteur, we can, in some cases, identify the infecting serogroup. And so you can see here that about one-third of the cases of leptospirosis in men and France are due to the heterohemorrhagia serogroup, which is responsible for the most severe infection. So what about leptospirosis in animals? So here in Lyon, there was a recent study a few years ago from the veterinarian school of Lyon, where they captured some rats, more than 80 rats, and they observed more than 40% of prevalence of leptospirosis in rats. So most of the rats, not most, but a high proportion of rats are carriers of the pathogenic bacteria, of the pathogenic Leptospira. The same group, more recently, also did a study on a large, they screened more than 1,000 samples from wild animals, so all animals except rodents, except rats, in the east, north, mostly north and east of France. And so they observed for the prevalence of leptospirosis in these wild animals. And unexpectedly, as you can see here, they observe that the hedgehogs are a good reservoir, important reservoir for leptospirosis. So the pathogenic Leptospira is present in many animals, and they are the reservoir and they're going to contaminate the environment. And here just a recent paper also where we showed that we identified with some colleagues in Belgium, where we identify several cases of leptospirosis due to the presence of pet mice, of pet rats, so transmission from these pet rodents that were positive for leptospirosis, and transmission to their owners, you know. So there was few cases of transmission from pet mice, pet rodents, pet rats to their owners. So this was the situation in men and France. So what is the situation in French overseas territories? Because this is also one of the missions of the reference center. So as you can see, so we are about 1.9 cases for 100,000 inhabitants in mainland France, but the incidence can be 5 times to 500 times higher in French overseas territories, in Martinique, in Guadeloupe, in the French West Indies, in Futuna, in New Caledonia, in the Pacific Ocean, French Polynesia, Mayotte, La Réunion, and French Guyana. So the incidence is much more higher in these French overseas territories. So what looks like the agent of leptospirosis? So this is one of the topic of research that we have in the lab, trying to better understand what are the variance factors of these bacteria. So again, so you can see these bacteria that are thin and with this helical shape morphology. They have a cell wall architecture which is very closely related to similar to the gram-negative bacteria, with 2 membranes, outer membrane and the inner membrane. And you have lipopolysaccharides at the surface of these bacteria. So these lipopolysaccharides are the major antigens, and the immune response is directed mainly against these major antigens. And so when you have endoflagella, so these endoflagella, there's one endoflagellum attached at each extremity of the bacteria. So as I told you before, these endoflagella are located in the periplasmic space. And the rotation of these endoflagella makes these bacteria highly motile, and they can rapidly disseminate in the organism, in the host, and reach the target organs. So just for you, for some of you who did not see these bacteria under the microscope, so this is a picture under dark field microscope. You can see these bacteria that are highly motile. And this is one of the topic of research that we have in the lab, is to understand how these bacteria move very fastly in a highly viscous media. So, as I told you, there's a high diversity of species and cerovars. So today there are more than 20 species that have been described. These species are clustered in 3 different groups. The saprophytes, which are non-pathogenic bacteria that are found in the environment. The pathogens, which are responsible for the most severe infections in humans and in animals. And there are a third group called intermediates, which are closely related, phylogenetically related to the pathogens, and that are probably responsible for milder infections in humans and in animals. So here this arrow in red, just indicating Leptospira interrogans, which is probably the species responsible for the most severe infections in humans. So there are major knowledge gaps in the field. So, for example, there are several questions, like, for example, what are the drivers of this emergence of the disease, as we observed in Europe and worldwide? Is it because of the climate change or any other factors? So this is some question we'd like to address, to answer. What is the ecology of the bacterium? So the survival of the bacteria in the environment is very important, is a key in the transmission of the disease from the environment to humans. So we don't know a lot about the survival of these bacteria. What makes Leptospira interrogans a major pathogen? So this is one of the topics also of the lab. What could be the effective control measures? So it's a complex cycle, so we can have intervention in the environment or in the animal reservoir or in humans. So there's many things we can try to do to control this disease. And what method also can be, there's also an urgent need trying to develop some new diagnostic tools to make an early diagnosis of the disease. Because what I did not say is that this disease can be treatable by antibiotics. There's no non-resistant to antibiotics known in these bacteria, but the antibiotic has to be done very early in the course of the disease. Otherwise, to be complicated to cure the patient. Just briefly to show you the course of the infection, so the kinetics of infection. So the incubation period is about 1 week, and then so you're going to detect the bacteria in the blood during the first week after the onset of the symptoms. So you can do PCR to detect the bacteria in the blood, and then after the second week, during the second week after the onset of symptoms, the bacteria will disappear from the blood, and you will detect in this case the antibodies against the bacteria, and so you can use different surgical techniques. Like an ELISA or the macroscopic agglutination test. You can also detect the bacteria in the urine after the second week after the onset of symptoms. So I think I'm going to conclude. So what are the challenges of this neglected but emerging zoonotic disease? So we have a critical need for diagnostics and trying to convince commercial partners to invest in this and to develop new tools for the diagnostic of the disease. This is particularly true in tropical countries, in poor countries. Most of the tools that we have today, they are not very efficient with poor sensitivity. We need also to isolate more strains to have a better idea of what are the strains that are circulating worldwide. I didn't have time to say, but these bacteria are fastest used bacteria, difficult to grow and difficult to isolate from biological or environmental samples. So we don't have a good idea of the diversity of the strains that are circulating worldwide, and this is very important to better estimate the efficacy of vaccines or diagnostic tools that we have available. Vaccine development, so there are some animals and human vaccines that are available, but they confer short-term efficacy, and they are usually serovar specific. Variance factors, so all the pathogenesis is poorly understood, and there are very few groups working on the disease and on the pathogen. Clinical investigation, trying to better understand what are the prognostic factors that are responsible for the most severe cases in humans, and trying to better understand the transmission of the disease. So with that, I would like to thank all the people from the lab at the Institut Pasteur. Thank you for your attention, and if you have any questions, thank you.
you.